time to start. So today, our presenter is Dr. Michael Strickland. He got his PhD from the Oldham School of Ecology at UGA, and he worked as an assistant professor at Virginia Tech. And he currently works at the University of Idaho in the Department of Soil, Water, and Systems. And today, he'll be presenting on the ecosystem processes in the age of antibiotics.
All right, so thanks, Rick. Thank you all for joining me here this afternoon and listening to what I have to talk about. So this work is like Jared said, going to focus kind of on ecosystem processes and kind of how antibodies might affect those things. But ultimately, I thought I'd give you kind of just a brief overview of sort of what it is that my lab does and sort of the direction and kind of how we came to this sort of antibody question. And so ultimately, a lot of this research was driven by this idea of microbes being this really important part in kind of biochemical cycling, right? And the idea that understanding how change in these communities, whether it's compositionally or biomass or what have you, right? How does that ultimately influence things like carbon and nutrient cycling at an ecosystem scale? And so a lot of the research that we've been doing around this kind of topic includes things like understanding how change in microbial communities can influence litter decomposition, how differences in those communities impact that rate, right? Some work looking at how the volatile organic compounds derived from leaf litter can also be utilized by these microbial communities, and also how maybe change in those communities influences the types of volatiles that are produced. We're also looking though at like these interactions with other components of the communities. So how trophic structure might influence the types of microbes that you have, how they might function, sort of like carbon nutrient dynamics within that. Excuse me. But we also have kind of some more of these more applied angles, right? Where we're looking at variation in sort of cropping or management systems. So things like cover cropping and cover crop diversity. And additionally, kind of coming out of this, right, and something that I kind of think has bridged both the sort of basic research and applied research, or some of this work that we're starting to look at in terms of these antibiotic effects, right, on microbial communities. And it's this last one that I really sort of want to focus on today. And it's some work that we've been doing for I'd say quite a while now. It's something I started as an assistant professor, and it's kind of continued on through my career up to this point. And this work is really starting to look at what I want to cover today is kind of this idea of what this antibiotic problem is and kind of set the framework for this, but also then start to dive into some of the microbial and ecosystem implications of it, in particular, looking at some of the research that we've done on trying to assess, one, if it's a problem and what that problem is. And then cover a future direction, and in particular, some research that's just recently coming out of my lab by one of my current graduate students. So when I was first kind of started kicking off this work, we came across this paper and this quote, right? And this idea that the only environments that are truly exempt from the influence of human antibiotics were those that existed before the antibiotic era, right? Before essentially the discovery of penicillin. And I think this is a pretty powerful statement. I think it's kind of like, it's almost like our I'd say our view of like microplastics today, right? That we know that these things sort of permeate our environment, and we know they're almost everywhere that we look, but understanding what the implications of these, right, are, I think is really important. And the focus though that kind of took away from this though is thinking about, well, where are a lot of these antibiotics being utilized, right? And so when we look at this, we oftentimes maybe think of it as a medical problem, that you're given an antibiotic and you're not using the full dose, right? And that it's being excreted or lost in effluent, right, from hospitals, if you will. But when you start to look at the numbers, something like 80%, right, of U.S. antibiotic sales wind up being for livestock, which is a pretty significant chunk. And in fact, the estimates of these, right, are going to increase by within the next 20 to 30 years. Particularly what we see is that developing countries, right, are actually going to start to see bigger increases in antibiotic use particularly because of the rise of middle class in certain places and an increasing demand for meat products. Within that, though, when we think about this, this winds up being roughly equivalent within the US to something like 33 million pounds of antibiotics annually, right, that are being given to livestock kind of across the board. Now, while that's a lot going into a cow, what we're really kind of interested in is like what's coming out the other end, right? So what are the amounts of antibiotics and what does that mean, right? So essentially what happens when these antibiotics actually enter the ecosystem, enter the environment that we're concerned with. And roughly when we look at this is somewhere around 13 to 31 million pounds of antibiotics that could potentially enter the environment a year, right? And this can be as kind of active antibiotic or as metabolites of antibiotics potentially. And it can vary, right? So these are just some, oops, what did I do? Sorry about that. So these can vary, right, depending on the type of antibiotic that we're looking at, right? It also can vary as how much of that is ultimately excreted, how much of it is actually, for instance, the sort of whole compound or active antibiotic versus a metabolite of that antibiotic as well, right? So those sorts of things can vary, and we understand that. It also is something that makes it kind of challenging to understand some of the implications of these, right? Because it can be difficult to actually track them in the environment as a compound at times. But luckily, I'm not focused so much on the antibiotic itself, right? But what are potentially some of these environmental costs of the antibiotic and understanding what those might be. And some of them that we kind of came up with were first this idea that, you know, maybe not surprisingly, if you have antibiotics going into the system, you might see that these soil microbial communities exhibit increased antibiotic resistance, right? So we might measure, quantify antibiotic resistant genes and try to better understand, are we seeing an increase in those particular set of these genes or not? You might also expect to see some shifts in the microbial community composition. For example, if you're adding like antibiotics that are targeting bacteria, which primarily is what we're focused on here, right? You're going to see that probably maybe fungi increase in abundance, for example, right? And then what does that mean in terms of how that shapes the rest of the community, okay? And then finally, this is what I think is what I've been really interested in is understanding whether or not the addition of these antibiotics can alter really how these communities are functioning. And in particular, does it lead to this sort of less efficient microbial community overall? And in particular, when we're thinking about something like carbon use efficiency, right? Or microbial growth efficiencies, things like that. And really, it kind of harkens back to these classic studies, right? On antibiotics that we might have looked at in microbiology, right? Where we have, excuse me, where we have like resistant organisms here, right? Versus a wild type or an organism that's not resistant to antibiotics here. And the idea, that resistance comes with some sort of metabolic cost. And is that metabolic cost ultimately scalable, right, to an ecosystem level? And so in order to start to kind of try to disentangle this, what we've done is kind of paired multiple sorts of approaches to look at and investigate this. So we're looking across like kind of doing these larger scale observational sorts of studies. We're doing some field scale, kind of like common garden experiments, and also some laboratory-based approaches. And so I'm going to kind of touch on some of these various studies throughout this talk. Sorry, I'm tested negative for COVID. I think I need to tell you that, but I'm also like coughing and anyway. So the first of these studies is actually this nationwide assessment that we kind of kicked off on. This was work led by Carl Webking, who was a graduate student, PhD student in my lab, where we had I think it was 11 or 12 sites across the US. You can see them as these little black dots here. And what we were doing is kind of looking at dairy farms on land grant institutions primarily across the US. And so we were comparing reference sites, so sites kind of outside of a pasture, to sites inside of a pasture where cows were present, right? And the first thing that we kind of looked at here was what was sort of the antibiotic resistant gene abundance within these sites, right? And so if you look here, you can see on some of these, for example, here, you can kind of see this little blue halo. That's just indicative of sort of the count, right? Number of copies of that antibiotic resistant gene. So these are the reference sites, right? These are the sites that we're not having like this kind of cattle exposure, if you will, at this location. And then when we looked at antibiotic resistance where the cattle were present, we saw that overall, right, there was actually a pretty big increase, right? So this is obviously making a highlight of that. But I think what's interesting here too is that, we don't always see increases, which is kind of interesting. Some sites actually don't increase a lot, some increase a lot, and who knows what this is attributed to. It could be management, it could be soil conditions, it could be a variety of things, right? But we also tended to see that when we looked at some of the various genes, we see that overall, where we had cattle present, they tended to be higher, right, than where we had the reference sites. Two of which were significant: AMP-C, which is resistance to like beta-lactam antibiotics, for example, and then one of the tetracycline resistant genes tended to be in higher abundance, right? Now, we also wound up looking at microbial community composition. So these are for bacterial communities assessed via 16S amplicons. And so this is a PCOA. If y'all aren't familiar with these, you're gonna see a bunch of them. So I'll give a brief overview. Basically, if points are closer together, that means those communities are more similar. If points are further away, for example, from one another, then those communities are more dissimilar, right? Or less similar to each other. And so overall, what we found though is that we get a shift, right, in microbial community composition where you had the presence of cattle versus where you didn't. And it was kind of driven by some major taxa. So for example, we saw an increase in firmicutes and we have a proteobacteria where we had cattle present. Some of these can be environmental indicators of ARGs. And in fact, I think what was interesting is we see this 25-fold increase in Acinetobacter, right, which is a pretty widely known group of bacteria, particularly with regards to their resistance to antibiotics. In fact, it's like one of these groups, right, that is a potential hazard, right? Now, this isn't like the main one that we see in hospital-borne infections, right? This is another identified as a potentially different species, but it's still kind of interesting that this is a group that we see pop up, right, in these soils. Now, this starts to get at, though, some of these ideas I was talking about with regards to sort of microbial efficiency. And in this work, what we were doing is basically calculating microbial mass-specific respiration, right? So we were quantifying both the biomass, the initial biomass of the soil, right? The biomass of the microbial community in the soil, or the microbes in the soil, and we were quantifying how much respiration was being produced from that soil, right? So basically, what we're getting at is how much respiration per unit of microbial biomass do we have, with the idea that the higher the respiration, right, the higher the mass-specific respiration, sort of the more active and more stressed potentially these communities are, right? So just note that the higher it is, right, it means that it's potentially less efficient overall. And so what we found here is that where these, oops, I feel like I'm in a weird position here. What we found here is that where these, where cattle were present, we had greater mass-specific respiration than where they were absent. And when we looked at relationships with basically antibiotic-resistant gene copy numbers, we see that where the cattle were present, we see this positive relationship, potentially indicating maybe that where you have greater resistance occurring in these systems, you may see that you have lower overall microbial efficiency occurring. Now, we worked this up though in kind of a back of the envelope way, so I hate putting this up here, but it winds up, something like a twofold increase in mass-specific respiration could potentially be a significant amount of respiration or carbon loss from the system annually, right? And so overall, from this component of the study, right, we were seeing that these inputs, these cattle inputs potentially were stimulating an increase in antibiotic-resistant genes. We see the shift in bacterial community composition. We also see this decrease in microbial efficiency occurring within these systems, potentially due, maybe to this relationship to the maintenance of antibiotic resistance within the system. But I think one of the big questions, right, is that obviously this is an observational study, and then trying to tie antibiotic use directly to what we're observing here is kind of tenuous, right? So it's a little tricky because we don't have a direct manipulation of the antibiotics itself, if you will. And so what we wanted to do is kind of see if we could get at that question, right? Could we actually influence kind of what antibiotics are going in and whether we can kind of control the actual inputs of those antibiotics to some degree. So this was a study that we developed on this, and this is more of this sort of common garden experiment, right? Where what we're trying to do is essentially look at how these kind of plots of soil are responding to these additions of antibiotics in a field setting. We labeled it the SCARE study. This is my grad student who came up with this, which is the soil carbon antibiotic resistance experiment, which I think is kind of fun. He was really good with acronyms. And what we wound up doing is going out there and adding manure or no manure to these systems across really, it continued for 3 years, but this is just that first year of this research that I'm presenting today. And what we had is a no manure control, and then we added manure from cattle that were either given no antibiotics, right, or were given one of 2 different types of antibiotics, cephalosporin or pyridomycin. Cephalosporin winds up being a bactericidal antibiotic, pyridomycin is a bacteriostatic antibiotic. And during the kind of peak growing season, right, of each of these years, we then conducted a 13CO2 pulse chase on these, where we added 13CO2 to these chambers, and then basically kind of tracked it through, right, into the above ground plant material, the roots, the soil, and then back out again as CO2, okay? We also wound up assessing microbial community composition again. Again, this is like after that first year. What we observed here, once again, is that where we had added antibiotics, right? So these kind of blue and yellow dots here, we tended to see that those communities shifted or were distinct from the communities where we didn't add antibiotics. So the no manure control and even the control manure plots. We also tended to see, there were some changes in various taxa. In particular, we again see that where we added cephalosporin, right, or manure from cattle given cephalosporin, we tend to see Acinetobacter increase again. But we also see some interesting things like with the pyridomycin treatment, like we see an increase in acidobacteria, which are typically we think of as probably case strategists, right, or slower growers in these systems. So they tend to increase in abundance with this addition of this pyridomycin, which I think is also kind of interesting. We also tended to see a shift in fungal dominance within these systems, right? So this was assessed via qPCR, but we see it particularly where we added pyridomycin and an increase in fungal dominance, right? Primarily what we're observing is that where you add manure, you increase fungi, right? And where you add antibiotics, you decrease bacteria. Maybe not too surprising, but it's kind of interesting, right? That we're actually able to sort of see this kind of wholesale shift in kind of the dominant organisms within these communities. Now, I think that if I were to present this to most soils biologists, right, you'd expect that if we're increasing fungal dominance, what do we think would happen? Would they become a more efficient system or a less efficient system? Anyone want to throw out a guess? Anyway, we'll move on then. I would normally say that we probably say a more efficient system, right? Because fungi are like the superheroes of the soil world, usually, is how I argue they are. But I sometimes disagree with that. And in particular, I think this study kind of highlights that maybe that's not the case, right? What we actually see is that when we look at respiration, so we track this across 7 days. When we look at cumulative respiration, we actually see that this pyridomycin treatment was respiring more of that 13C label, right, over that 7-day period. And that the control manure was actually respiring the least amount, right, or those plots associated with that control manure were respiring the least amount of 13C. We also tracked it into the mineral-associated organic matter. And so what we observed here is that when you look at pyridomycin, right, we're getting less 13C recovered in that mineral-associated carbon fraction than we are in actually the control manure. The no manure control is a little variable, right, in this instance. And when you look at this kind of as a overall system efficiency, right, what we're observing here is that when we look at the ratio of how much of that carbon was fixed versus how much was respired, we see this pyridomycin treatment falls out as being the lowest, right? So overall, less of the carbon that was fixed was actually retained within the system, right? More of it was lost from that system ultimately. Now, overall, right, what we're seeing here is that these antibiotics wound up leading to kind of what I would argue fundamental shifts in these communities, right? Maybe less dramatic when we look at those community compositional changes, but particularly when we think about fungal bacterial ratios, we see pretty big changes, right? We're also seeing that they lead to this decrease in ecosystem kind of carbon use efficiency, right? Suggesting that these antibiotics may have an implication for how carbon is cycling within a system. And also, I would argue, though, that manure is not necessarily bad, right? It just depends on how you're managing those cows and the kind of manure that, where that manure is coming from. In fact, we're seeing in this system, right, where you have manure from cows not treated with antibiotics, we're actually getting higher kind of ecosystem carbon use efficiency, right? And it's probably because, you're stimulating plant growth and you're alleviating stress on the plant, so the plant can fix more carbon and retain that carbon. But you kind of wash that out when you add those antibiotics, because then you're disrupting that microbial community. Now, I'd like to highlight this because this was a fun thing. So Carl, my grad student, got quoted in Scientific American for this, and these are like one of the highlights. Like I've always wanted to like have something in Scientific American, and like Carl got it, and I was like, sweet. So, but this is just one of these things, and so Carl was going out there and actually looking at this system over the course of this 3-year period, approximately, right? And he was actually monitoring respiration weekly, right? He was also, admittedly, he was adding manure like weekly, right? I always joke that he was acting as the back end of a cow, but he doesn't appreciate that. So I only say it to y'all. But he was going out there monitoring that. And one of the things that we picked up on here, and we thought was kind of interesting, we're still kind of investigating though, is that early on, right, we see that like this pyridomycin treatment has what looks like a much stronger temperature response, right? So it looks like there's more respiration at higher temperatures in comparison to the other treatments. And when you kind of like pull all this data together and calculate Q10 values for it, we actually do see that pyridomycin tends to have a higher Q10 value, right, than do the other treatments, which is sort of interesting because here you have this thing that we're not manipulating temperature here. We're just like modifying kind of the community with these antibiotics, and we're altering the temperature response of these communities. And it was from this work, right? We've actually done some follow-ons with this. I'm happy to discuss later, but I don't want to go into too much detail about it. But from this work, it really made us start to think about, well, are there potential interactions occurring between temperature and antibiotic treatments, right? And what do those look like? And there's been kind of a big kind of push, right, for looking at multiple kinds of global change factors and the combinations of those, right? Not looking at just like an individual sort of temperature effect, right? But how do all these other factors play together to influence it? And so a postdoc in my lab, Jane Lucas, basically ran this study where she was looking at 3 different antibiotic treatments. We had a control and then a high and a low dose of Menincin. So Menincin is an antibiotic that's typically given actually in beef cattle supplements. It's one of these antibiotics that is like USDA approved, essentially. So it's not typically for human use, but we thought it would be interesting to kind of look at in terms of these sorts of studies and implications for soil communities. We also looked at basically maintaining like these microcosms across 3 temperatures, right? So 15, 20, and 30 degrees C. So just a big range of temperature to see if there's going to be any effect or mediating impact of antibiotics, right? Kind of had some general expectations that, temperature, obviously we're going to see this increase probably in the short term of microbial activity and respiration. Whereas with antibiotics, you might expect that, you might decrease microbial activity and respiration, but we also know that we might get these changes, right? Because of both of these in terms of like microbial community composition or fungal bacterial ratios as well. Now, what we observed here was that, again, where you add antibiotics, right, particularly bacterial specific antibiotics, we see this increase in fungal dominance. And in particular, we're seeing like, at 20 and 30 degrees is where we start to see that change in fungal dominance occur, right? At lower temperatures, it's a little crazier, a little less dramatic, right? And so again, like we're starting to see, a common pattern. Maybe not super surprising. We used to add antibiotics, right, to assess like the contribution of fungi and bacteria in the system. I think there's some big questions about whether that is actually a really useful way to do that for lots of reasons. But we're starting to see that there's the implications that these antibiotics can have kind of these really big changes or shifts in the community that we're seeing. And I like this study because this is one of the few ones where you're doing microbial ecology and you actually kind of see effects, right? And so in these jars, right, where we have the control, the low dose and the high dose, I believe these were the 30 example of a 30-degree treatment, but you can actually see, kind of these hyphal-like growth on the surface of these treatments, right? And where you have the no antibiotic addition, right, you don't see any of that, which I think is kind of fun to observe. Now, from a community composition perspective, we're seeing that for the bacteria community, right, its composition is dependent on both temperature and antibiotic addition. There's actually this interaction that seems to be occurring. And what we observe is that you'll see that, basically we have 15 degrees, 20 degrees, and then 30 degrees. You're getting a shift in the community along this axis, right? But you also have distinct communities, right, that are sort of these controls up here, so the water only. And then you have the bacteria that have been given antibiotics down here, right? Or the communities that have been, the treatments that have been given antibiotics down here, which are very distinct from these. With fungi, we're not really seeing this antibiotic effect, but we're still seeing a temperature effect, right? So on one hand, we're seeing, that when you start putting these multiple global change kind of factors together, different things are responding sort of to different treatments, and some are interacting and some are not. And honestly, I'm not sure 100% what to make of that yet, I'll be honest, but I think it's interesting because it just makes it that much more kind of a higher level of uncertainty in our expectations of what's going to happen in these systems as we invoke more change. Additionally, when we start to look at mass-specific respiration again, what we observed in this, again, this is a short-term laboratory-based experiment. What we found is that, with these doses of antibiotics, and it didn't matter if it was a low dose or a high dose, we tended to see this increase in mass-specific respiration. But with temperature, right, there was a really non-significant effect on mass-specific respiration, although it does look like it may decline slightly as you increase temperature in this instance. Again, it was a short-term study, so we might expect that to change, obviously, as we complete soil gardening or get some more changes in these communities. Now, to kind of bring this section home, antibiotics wind up altering these temperature responses of these communities, right? That's kind of what we were seeing in that initial kind of field-based study, which we're still following up on, right? Which I think is pretty interesting. It looks like antibiotic and temperature interact to shape microbial community composition, but then it's kind of like these independent effects, right, or additive effects potentially on things like mass-specific respiration. And then antibiotics wind up, again, kind of decreasing this microbial efficiency. And I say again, like even with this increase in fungal dominance, right, which I would not always expect. Typically, I think where we have, again, more fungally dominated systems, sometimes I think more efficient systems. And it was this kind of question, right, that kind of led to some of the future or kind of current work that we're doing on this topic, where what we wanted to do is really assess whether these antibiotics are having kind of differential effects in a system that is more fungally dominated versus more bacterially dominated, right? And so what we wound up doing is going out to basically a forested site, right? A site that we expect to be more fungally dominated, and then remnant prairie sites that we expect to be more bacterially dominated, right? So, and looking ultimately at how antibiotics potentially change the way that these systems function. Additionally, what we wanted to focus on here were not so much the large doses that we might expect with like a agricultural sort of input, but more what I would think of as the windborne or the dustborne sorts of concentration of antibodies that you might expect to sort of permeate, right, kind of across a landscape. within these, we wound up focusing on 4 types of antibiotics. We had oxytetracycline, tetracycline, cefepime, and penicillin. And so these were combinations of both bactericidal and bacteriostatic antibiotics, and also kind of naturally occurring antibiotics and semi-synthetic antibiotics, right? So the idea is that you have these natural antibiotics like penicillin and tetracycline that probably some of these soil microbes have seen before, right? And then you have, some of these antibiotics like cefepime or oxytetracycline that are semi-synthetic that maybe, you know, these microbes in kind of some of these less disturbed systems have had less prior exposure to as well. Now, just to give you an instance that, you know, at least our sites that we picked are different in terms of their fungal dominance. This is the forested site, right? So this is, again, the acute PCR assessment of fungal dominance. More fungi, less fungi, right, in the prairie system. And so based on that, right, we were expecting to see potentially stronger effects of antibiotics, right, in the system where you have more bacteria, right, particularly if we're targeting bacteria, than in the system where we have primarily fungi. So we basically took these soils and we exposed them in a jar, right, to antibiotics, direct doses we have high and low concentrations that sort of bracket the ranges that we expect might be inputted into these systems via kind of like, you know, windborne or dustborne inputs. And so then after 60 days, what we see is that this is change in the fungal to bacterial ratio. And so really what you're looking at is, you know, here are our controls, so no antibiotic additions, and then we have our whole range of different antibiotics that we added here. What we found is that, at least in the prairie system, the one in the kind of orangish color here, orangish red, is that we tend to see a more marked increase in fungal dominance, right? I would argue, than what we observed in the forested system. So the antibiotic inputs there are definitely like shifting, you know, these communities in that way. Now, what I found kind of interesting here, and this is again looking at mass-specific respiration, and then this is actually 13C biomass to 13 CO2, right? So we did a little glucose pulse chase in these. So we have 13 CO2 to 13C biomass estimates here. What we see is that, you know, in a lot of instances, when we look at these treatments versus the control, on average, we tend to still have higher mass-specific respiration, right? Where we've added antibiotics, primarily occurring with things like cefepime, in this instance, and then cefepime in the high and low doses over here, right? And arguably, though, right now, obviously these differ in terms of you know, there's a difference in terms of the prairie and the forest overall in terms of mass-specific respiration. But I'm not seeing like necessarily a big marked change between the two, I would argue, right? And honestly, like, while we're still seeing these big shifts in the community, right, potentially, we're not necessarily seeing that's necessarily indicative of what we're observing in terms of mass-specific respiration. That they tend to, you know, antibiotics in both of these systems, even at low doses, tend to have this kind of negative effect on microbial efficiency. But what I think is kind of interesting that Dan Du, who's the grad student working on this project currently, pulled out of this, though, is when we start to look at these by the type of antibiotic, and you can see this both in the forest and the prairie, that we tend to get higher mass-specific respiration where we have the semi-synthetic antibiotic, potentially because, again, these organisms haven't been exposed to this previously or haven't had as much exposure to this type of antibiotic. Then when we look at the control or the naturally occurring or more naturally occurring antibiotics. We also find, though, that at least in this short-term study and at these low doses, that we tend to get a greater response, greater negative response in terms of mass-specific respiration in these bacteria with these bactericidal antibiotics than we do with the bacteriostatic, which I also think is potentially interesting. It highlights that, you know, we need to pay attention to potentially the types of antibiotics that are going into these systems and where they're coming from as well. In conclusion for this section, fungal dominance, right, may shape some of these community responses. These antibiotics type and action may actually play a pretty important role in determining the ecological consequences of their addition, right? And I think even from like management perspective, we may want to start to consider what that is, right? And how do we manage the use and the types of antibiotics that are employed in systems. And then, it's again that it seems like even at these very low doses, these antibiotics tend to decrease microbial efficiency within these systems. And so to bring this kind of home, antibiotics wind up being everywhere, right? Antibodies, I think, have the capacity to shape soil microbial community composition and function. We see that pretty consistently across a variety of scales and a variety of systems. And ultimately, I think may have this potential to kind of shape the response of these communities even to other global change factors. So things like increasing temperature, for example. And so that's about all I have for antibiotics. I thought I'd make a pitch really quick that we have this new facility that we're developing at the University of Idaho. And if you're all interested in it, feel free to chat with me about it. But basically a focus on looking at deep soils in an ecotron kind of setting. And with that, I'd be happy to take any questions. Thank you all. Oh, yes.
Very interesting. So you pointed this out, but most antibiotics come from soil, right?
Right.
So they've been made by bacteria or fungi present in soil. There's some question as to what their biological roles, I guess,
Right.
be.
Right.
Or are they produced with the intention of clearing out your immediate vicinity, such that you can compete for resources? Or are they in fact used for a signaling molecule?
Right.
That we don't really know what the purposes of these things are.
Yeah.
And so, like, whether or not a bacteria gets exposed to what we call inhibitory concentration of antibiotics can actually impact whether or not, or what the ultimate outcome is going to be for that bacteria.
Right.
So, I guess, like, by way of a question, do you think that these levels of antibiotics are inhibitory, or do you think that they are perhaps doing something else that might invoke a different response?
Yeah, I think that's honestly an awesome point. I think the antibodies that were potentially in some of the studies, right? For example, I think that they could be inhibitory, right? We're seeing, for example, I think where we're seeing like maybe these declines potentially in things that are indicative of bacterial biomass, like 16S copy or something like that. Like when we're getting those manure inputs potentially, right? Those might be a high enough dose to actually be inhibitory, right? Now that last study I showed though, it's interesting to me because we're seeing like, usually you think of a dose response, right? We think that sometimes we see the stronger effect where we add a higher dose. But again, these are really low concentrations, right? I have to look at my notes again to remember exactly what they were, but they're like orders of magnitude lower than what you would see in a direct like cow patty input. But sometimes you're seeing like where, and it's in this study as well as a few others, but we're seeing like the low dose of that, right? The one that's like 10-fold lower than the high dose is actually having a stronger effect than the high doses, right? And it makes me wonder, yeah, exactly that. And if we're seeing some sort of non-inhibitory change in how those communities are functioning, if that makes sense at all. And I agree, I think there's a play at both of those ends. And additionally with that last study I showed you, we've been we have, I didn't put it in here because I thought it'd take too long, but we have microbial community composition data for that as well. And we're not seeing like, what we're not seeing is changes in the community composition itself. So we're not seeing shifts in like the centroids of those communities, right? They're not distinct. But where we're adding those antibiotics, what we are seeing though is greater, it looks like greater kind of dispersion, right, around that centroid. And so you're getting these more random communities within that community space because of those additions of antibiotics. And I wonder if that's indicative of something like those signaling effects, right, that are going on. And I think working at both ends of those spectrums has been interesting. And I agree, I think it's both are happening. It's just what matters and when does it matter has been a big kind of question that we're starting to tease out, I hope. That's a roundabout way of saying, I don't know that I know the answer, but I kind of have some indications of directions to go.
Interesting, thank you.
Yeah. Yes.
So a plant focused question. So some of the work that shows that there's a shift towards more fungal dominance in these communities. So there's good fungi like mycorrhizae, for example,
Yep.
and there's pathogenic fungi, and there's fungi like they can help decompose things, which is good, but only to a certain point. Because then if they decompose too much, you don't have carbon stored in soils anymore. So is somebody like, or is you or somebody else looking at like, if the fungal community, if it's shifting towards more fungal dominance, like what's happening in that world?
Right, right. And we do have some, we have data on that. I have ITS sequence, ITS sequences for the composition of those communities, and I'm trying to recall. Do we have that for this? See, this sounds horrible. I'm trying to remember exactly all that data. And I was trying to remember because we were kind of engaged in looking at that because we were like, well, what's happening here? Because is it because maybe we're getting like higher AM, dominance in here, or what's the composition? And for some reason, I'm recalling that maybe we didn't, at least with the ITS sequences, we didn't see any major differences between the composition of those communities. Now we saw that the dominance increased, right? And I think it's indicative that we saw probably what was an increase in fungal biomass. But we kind of see that like fungal composition sort of stays the same, but there's just like maybe more fungi. And I'm not sure, you know what I mean? Like, I'm not sure how quite to disentangle it, because our original idea was, you're seeing this increase in fungal dominance and you're seeing this decreased efficiency. And I've seen some arguments, that maybe you're getting these less efficient kind of cycling of carbon because of like AM fungi or something, right? And I'm not sure that I have evidence necessarily for that, but I don't know, I'm talking around in a circle, I think, but.
Yeah, it just seems like that's kind of in terms of ecosystem function, like a cool area. Not like you don't have enough on your plate already, but.
And I think even like looking at, we have more data too on like the systems that we're looking at currently that are like the prairie system and the forested system. And so we have a little bit more data on those that we're trying to dig into and get at as well. It also makes me interested in thinking about like some of these fungicides and things like that too that are being used and but I think we're gonna stick with this for right now.
Can I ask a follow-up?
Yeah.
So were your manure sources, were those from feedlots or?
Right, so the manure sources for that second study that we had was actually taken, we had basically what was an animal experiment. We partnered with dairy scientists on this, and so they were like, I think it was like 3 cows that were basically put in a pen, and we collected manure and urine directly from those cattle over the course of, I think, a week after administration of antibiotic, right? So we had control cows and then the cows that were each given those antibiotics. So that's manure, that's fairly well controlled. It was quite the experience of collecting it and then storing it because we got all of it kind of at once for a year. And then I don't think I made people happy on my floor because I kept it in a big chest freezer and it didn't smell good.
So does it only come out in the manure, or what about the urine?
Yeah, I think actually you see a lot of it can be excreted in the urine. So that we collected both, right? So we had urine and manure mixed. So it was kind of a slurry,
Oh, okay.
if that sounds
Okay.
interesting, yeah. And then we bagged it up in Ziplocs and then we froze it.
Okay,
And
right.
then as we needed it, right, we take it out to the field and apply it.
So did you measure chemical constituency within the slurry?
Yes, we did. And what's interesting is so in particular, you can see there are actually some indications again, like that subapparent treatment was having an effect, right? In some instances, right? But what we found is that you couldn't really detect subapparent in the manure or that slurry, right, after the fact. We were able to detect acrylomycin in there after the fact. And then we looked at like just general like carbon and nitrogen ratios, right? And there were no real significant differences actually between those two. What I would say though is like some of these antibiotics do act as a kind of diuretic. So I'd argue that some of the slurry was probably wetter than other slurry was, right? So in hindsight, we're sometimes like, I think it's a really, I still advocate for the way we do it, but sometimes it would have been easier to just like add antibiotic, right?
Yeah.
You know?
Yeah.
Well, in a lot of ways, it would have been easier to add antibiotic, but then you would have missed these manure effects and we wouldn't have seen, I don't know what that would have looked like though, too, like where you have those really kind of positive effects of manure only on that plant community, because nothing else was being added to that site, you know?
So like, what's the long-term persistence of the antibiotics in the soil environment? Like, are they absorbing the soil minerals? Can they hang out for a long time and like cause a problem later on? Like,
That's a good question, and one that I may not have the best answer for. My understanding is that some of them, you know, depending on charge, they can be, they can stick around for a while, right? I think some of the problems that we've run in, i.e. people who are also working on this project, is that like when quantifying this, sometimes you're looking for that like compound, right? And like, for example, like you can find it sometimes, right? And you're like, oh, well, it's there, that's great. But a lot of times these things are being like partially metabolized. And so you have to look for all those sorts of metabolites of that compound too. And some of those have varying levels of activity.
do they also have antibiotic
Yeah.
characteristics too?
Some of them can, right? And so some of those can be like these kind of sub-inhibitory effects as well, right? Which can also be problematic from a development of a resistance gene as well, right? Or development of resistance in the system. And so, you know, that's not my speciality, I guess I would say, but it can be problematic in trying to actually quantify how much is in there. And I know folks who have like done this even on like dairy farms, and you don't see it, but then you go out and you measure antibiotic resistance in some of these, and you see like high levels of antibiotic resistance. And so it makes me question as to like, what are we measuring when we do that? But I guess that's a roundabout way of not answering your question too. But yes.
I'm just curious, so in addition to antibiotic treatments, the cows receive probably some antifungal treatments, right? So if they're excreted together with antibiotics, then you have kind of interaction that prevent growth of the fungi, right? And the bacteria maybe even expect those are.
Right. And that could have been an effect, I think, probably in like, you know, some of those first studies that we looked at, like that observational study. The one where we were adding the manure, they had no previous treatments up until we, you know, they implemented those. But I agree, like, I don't know, like, it would be interesting to understand that. And then we also see, you know, some of these, one of the big things that we'll pick up on too is like, they'll give, depending on the system, but they'll give the cows like the, what is it, like the copper sulfide foot baths and things like that, which obviously heavy metals have antibiotic properties and stimulate antibiotic resistance and all that kind of stuff too. And we haven't even delved into some of that at all.
And you have nanomaterials that are entrants that have also antibiotic properties.
Right,
So
right.
it's kind of like, I wonder if you suppress the growth of the fungi, what happens to the microbial
Right.
communities? That's specifically for antimicrobial resistant humans.
And I would say the one thing that, you know, like from the bacterial perspective that we're seeing, which I wouldn't always have thought, which I wouldn't always attribute to kind of these changes in efficiency that we're observing, but we're getting these shifts in the community, and this is multiple times in these studies, the bacterial community toward what I would consider like more of your like oligotrophs or kind of case strategists, right? So we're seeing like increase in the dominance of like Acidobacteria or Verruca microbia and some of these other groups in the soil, which is interesting because I think that, you know, you're suppressing those fast growers, right? That probably take advantage rapidly of carbon, right? Well, we may think that they're not as efficient, but they're able to like capitalize on that carbon input, right? So they're able to grow quickly, like your proteobacteria, some of them, well, not all of them, but some of them, right? Are able to rapidly grow and capitalize on that. But you're disrupting that mechanism in the system. And while you're getting a shift toward what we oftentimes think of as these slower growers, more efficient kinds of groups, I think you're losing carbon potentially, or there's some inefficiencies in there because you don't have those faster growers, or they're not able to capitalize on it as much. So anyway. Yes.
So the slide that you showed where the increased fungal growth, that was in your temperature study.
Yeah.
So is that because it was controlling the bacteria and allowing the fungi to sort of assume the niche? Or was it stimulating some of the fungal growth directly, or can you separate that out?
I don't know that I can separate that out. I haven't thought about it necessarily that way. I've always thought about it like you're probably freeing up some of that niche, but it could be a combination of both, right? Because, well, the funny thing is like what we see is, so with the bactericidal antibiotics, you might be like, that fungi might be feeding on some of that bacterial biomass, right? That dead bacterial biomass, which could be a reason why you get bloom. We actually, sometimes we see some stronger effects with some of the bacteriostatic antibiotics, which are essentially kind of inhibiting growth, right? So it's not that they're necessarily leading to cell death, but they're definitely inhibiting that organism from growing, right, in that system. And so you're probably getting, in that instance, I might expect maybe a little stronger, just kind of the fungi exploiting that empty niche, right? And the bacteria unable to take advantage of it in that system. That would maybe be my expectation. But yeah, I don't have clear evidence, I would say, for it.
Don't you think it's weird you don't see differences in your fungal composition?
It is.
That is really weird, Mike.
I see, but we see it consistently. We've seen it in like multiple studies that we don't see huge changes in fungal community composition. I mean, we see like changes in fungal dominance, right? And you'll see like increases in ITS counts, but you don't necessarily see that. I don't have a really great explanation for it. I would think they're more connected than we think, than it observed, but then what we see, but it's almost like the fungi are just like increasing in abundance
But how can they all?
proportionally.
But how can they all of them be?
I don't know.
increasing? That doesn't, I would need repeat data to convince me that that was true, but it sounds like you have it.
Yeah, I mean, we see it in like both the field study that I showed you, we've seen it consistently in actually 2 laboratory studies now, that like the fungi compositionally stay relatively the same, or at least that we can't detect that change, right?
It's weird, man. You would assume some
Yeah,
of them would.
But we see these really strong changes in bacterial community composition.
Yeah.
I don't have a real good reason.
Yeah.
I guess.
Do you ever do these on bare soil?
Okay.
Like if you just compared like bare soil manure plus minus on bare soil?
Well, we have, I mean, some of the lab-based incubations don't have plants, right?
Yeah, all right.
And we actually have some studies that are, we have some other work coming out of some build sites, but those are problematic because we deal with like a summer drought, right? And so, anyway, yeah. Other questions?
I was going to ask the same, or if you looked at responses with changes in the plant community, like for example, more or less legume,
Oh, yeah.
in case the changes in the bacteria community could be for bacteria that are interacting with plant roots.
Right, that would be actually really interesting because we, kind of unrelated, but we have some work going on in southern Idaho, which has this huge dairy industry, right? I've been interested in like incorporating like the antibiotics, but we're doing like some cover crop trials in that area too, like various types. And I wonder if we could get at that. That would be interesting, actually. We don't necessarily, did we really look at it? I mean, this was like a fescue.
Don't downplay fescue.
I'm not
That's
trying to.
fine.
I'm saying though, it was pretty not like diverse, right? The build site we had. I mean, what was interesting from a plant perspective, I feel like, if I recall correctly, right, and we do have biomass numbers, I could go back and I'd have to look at this, but like we saw like, I mean, the plants just like, they were like, give me manure basically, and you know, they seem to grow well, right? But when we looked at like these impacts below ground, that's when we started to pick up some of these other signals. The piece I don't put in here, which actually is in the manuscript, though, we do see differences in because we did a 15N chase too, and I don't put it in here because it's difficult to like get at, right? Because we see differences in that allocation of nitrogen, I think, in plant roots and above ground plant material. depending on these antibiotics. And I think if I remember correctly, what happened was actually in that pyrrolomycin treatment, we're actually seeing greater 15N in the above ground plant material than we are in the other treatments. But I don't know if it's because we're knocking back bacteria in competition for that, because we added it as ammonium nitrate, and whether you're seeing less in microbial biomass, because we didn't actually look at microbial biomass nitrogen. I guess we could, we had those samples, but I just haven't looked at it. So we do get those different dynamics happening there. But I have to admit it, keep it off because it's difficult to put all that together. But I would expect under those instances, you would expect to see a shift, right? If you had enough time, maybe in plant community composition or something like that, which would be interesting. But it'd be interesting to look at it like, yeah, if you had a legume or a legume dominated system, what would happen too, right? That's cool. I might still. Should I check online?
Yeah, we have like 2 minutes left.
Okay.
So let's see if there's a question
Any questions online?
on Zoom chat.
Nope.
All right, well, if that's all the questions. Thank you very much.